Well, good evening. Welcome, everyone. It's lovely to see so many people here. So my name is Nick Fox. I direct the Dementia Research Centre, where I have the privilege of working with Dave. And I am also one of the heads of the Department of Neurodegenerative Disease at UCL. So it really is my great pleasure and honour to host this inaugural lecture for Professor Dave Cash. And we can have a little round of applause every time Professor is So it really is great to see so many colleagues from ION, from CMIC, from UCL wider, colleagues and friends, and some colleagues who we've worked with and have made a bit of a journey back here. Some from south of the river, which is, really quite a long way in London terms, but some from Sweden, some from the US. So I would like to very warmly welcome all these external visitors who've joined us tonight. But most importantly, and on behalf of us all here, I would like to welcome Dave's family. And I think, Annette, you get, as Dave's mother, get the prize for the longest journey, unless anybody wants to be coming from, maybe anybody from Tokyo here? They're still late to come in, so, but you get the prize. But Annette, thank you for very much for joining us, and welcome. Though having previously had you attend a DRC bowling event, I don't really think you get called an external visitor. You're sort of an honorary member of the DRC. Also wonderful to have Helen and Claudia and Charlie. Nice to see you here. That's really great. And also Pete and Sydney. I think I know that's very, very special for Dave, and I think it's lovely. So this is a very special occasion. An inaugural lecture is where we come together sort of at an academic level to celebrate an individual's academic, professional achievements. But it's also an opportunity to find out a little bit more about that person. No spoilers there. And an opportunity to look at that journey that takes people to this point. And there are very few opportunities for any of us to review and to celebrate that journey, that series of obstacles, hurdles, and achievements that take you through. All too often, it's only at sort of people's funerals. Not that I'm planning anything, but that's when people, say the nicest things about you. And so this is one of the rare exceptions to that. You get a chance to think about, reflect, and to share with us that journey. And those are considerable, your contributions that have been along the way to the field, to others, and to this institute, and hopefully to contribute to the culture as well as the achievements of a place, so that we make it the place where we would all want to work. And on a personal level, your contributions to the Dementia Research Centre and to me have been profound. But Dave's academic journey ranges quite a way from his prestigious biomedical engineering degree from Duke, and then a PhD also in biomedical engineering from Vanderbilt, before coming to the UK, very luckily for us. Apparently that's because he's Anglophile, or was before he got here. So that definitely, but I think it was mainly the music rather than the science, but nonetheless. And so he came to do a postdoc at CMIC, and it's great, Danny and others from CMIC are here. And he then spent a time in industry, and one of the things that Dave has brought to UCL and to the field is this ability to see things both from the perspective of academia, but also from industry, and hopefully to share that across there, which is not usual. So he worked for IXICO for a while, and there he was helping to run imaging for clinical trials before returning to UCL and joining both the DRC and CMIC in a joint appointment. Dave's track record is impressive. He's co-authored 200 papers, I think he's lost track a little bit, in some of the most highly regarded journals in our field. and he has been awarded highly competitive grants, most recently being awarded a Dementia Research Leaders Fellowship from the Alzheimer's Society. That's a grand and impressive title, so a Dementia Research Leaders Fellowship. He's supervised 10 PhD students to completion, at least one of which is watching online. Hi, Will. He's given multiple talks, nationally, internationally, and I have to say it was a particular pleasure for me and a huge recognition of Dave standing in the field when he was invited to give the keynote opening lecture at the Alzheimer's Imaging Consortium. Very popular, but it was to review the whole of the imaging field for that year, which is quite a daunting task, and it is a measure of the respect in which he was held to be asked that, and it's quite a challenge. He has and continues to have had really important leadership roles, just been invited to serve on the Council of iStart, overseeing the programs of the most important society and conference in dementia. But closer to home, Dave has been an incredible mentor and supervisor and a generous colleague with multiple contributions to the DRC and beyond. And being promoted to professor with tenure is an incredible achievement and a great honor. And it's certainly richly deserved by Dave. So please join me in welcoming Professor Dave Cash to deliver an inaugural professorial lecture.
Well, Nick, thank you for the kind introduction, and thanks to all of you for coming out tonight, and also those of you who are watching online. I'll say hello to you as well, Will. I can't fully articulate what an honor this is for me. I remember during lockdown mentioning the whole concept of an inaugural lecture to Helen and my kids, and thinking, gosh, one day I would really like to do that, and it's hard for me to realize that this day has actually come. I realize that I haven't had a traditional journey to this milestone, so I'm extremely grateful and humbled that the Institute and UCL have recognized my contributions to research. Also, as a disclaimer, I've been trying to get over the last vestiges of a cold for the last week or two, so I hope not to have any coughing spells along the way, but apologies if I do. These inaugural lectures are a wonderful tradition at the Institute of Neurology. A lot of my friends and family have asking me, what is an inaugural lecture? And so I thought about it, and the first thing that popped into my head was from my childhood. And I said, well, you know, it's kind of like an academic bar mitzvah. And since so much of my work involves measuring how the brain changes over the years, I thought it would only be appropriate to show how much I have changed since I last underwent a similar rite of passage. Though I have to say, you know, I lived in Shoreditch for years, and that outfit is far more home there than anything I ever wore while I lived there. So before I talk about my research, I first want to recognize all the talented people that make these studies a reality. First and foremost, I'd like to acknowledge the study participants. They selflessly give so much of their time undergoing complex memory tests, lumbar punctures, imaging visits, many other investigations. Thank you for all the trust that you've put into us and our research. And without your participation, there would be no data to analyze. Ann Parnell and Susie Parker do an incredible job navigating all the complex financial, administrative, and ethical aspects of our grants and our studies. To our fantastic research nurses, raters, study managers, and research assistants who make these studies run smoothly day in and day out. And to Fade and Narutsas and the research software engineers who keep our research IT platforms on the road and that they're robust and that they're fit for purpose. As Nick mentioned, it's important to note that I'm not the first PhD in my family. That honor belongs to my mother, Dr. Annette Cash, who's here, and who's a highly accomplished educator and researcher. She was completing her PhD in Romance linguistics when I was born, and her research has been wide-ranging, from medieval illuminated manuscripts in Galician Portuguese to the literary works of Cervantes, to some of the earliest uses of computers in Spanish education. And now she's investigating the use of generative AI for translation, particularly around medical conditions such as autism. Her love of languages did transmit over to me, albeit in a slightly unexpected manner. Instead of Spanish, Italian, and Portuguese, I opted for C++, Python, and R. The love of math, science, and technology came from my father, Claude Cash. I didn't have a lot of friends growing up, but I could always count on him for fun and companionship. When he wasn't being silly or indulging my every whim, requesting his attention, he worked with numbers and figures all day as an accountant. And here I am helping him double-check his figures. I was endlessly fascinated by any electronic device growing up, whether it was this huge transistor-based calculator from the 1960s that Dad kept in the basement, video gaming systems, or my first PC. I would just spend hours poking and prodding them, finding out what they could do. When I was 11, we moved from Columbus, Ohio in the States to Douglasville, Georgia, which is a small city about 20 miles outside of Atlanta. My parents' love of learning and how much they prioritized my education was evident by the fact that they helped create a brand new school from scratch when my favorite science and math teachers were leaving my current school. It was a tiny school. I want to point out, this isn't a form or even one class. That's the entire school right there, teachers included. And thanks to the continued generosity of my parents and grandparents, I was fortunate enough to attend Duke University in North Carolina to study biomedical engineering. Duke is known for its academics, its excellent research, and its hugely successful basketball team full of future NBA superstars. And the team was so popular that students would camp out for weeks to get the best seats in the house. But Mom, I can assure you, I didn't do that much. There were late nights in the lab working on electronic circuits and medical devices, and also my first research project. I had an undergraduate research fellowship under a professor named Wanda Krasalska to study the electrical properties of heart cells. But it almost didn't happen. As I was applying for this fellowship, I had a major exam in her class, and within 15 minutes left, I was absolutely sure that I was going to fail it. But somehow I rallied, and at the end of the day, I did well enough that she offered me the position. So doing that research project convinced me to pursue a career in medical research, and I moved about 400 miles west to Vanderbilt University in Nashville, Tennessee. I did my PhD under Professors Bob Galloway and Michael Miga, who were important mentors throughout my career. And my PhD was on image-guided liver surgery, where we would use MRI and CT scans taken before the operation to provide interactive navigational aids to the surgeon while they were trying to remove tumors from the liver. So as Nick alluded to a little bit, outside of my PhD, I spent a lot of my time as a DJ at Vanderbilt's radio station, WRVU 91 Rock. College radio in the US had a massive influence breaking new bands, particularly before the days of Spotify and Apple Music. Our broadcast would reach into 3 states over an 80-mile radius, meaning that we had some interesting diehard fans. I was music director there for 2 years, determining what music would be played at the station and getting to interview a load of great bands. And I also helped organize the celebrations for the station's 50th anniversary, including I was responsible for booking a full-day concert on the main lawn of the campus. About 6 months after my PhD, I took a postdoctoral role at UCL at the brand new Center for Medical Image Computing with Dave Hawkes and Derek Hill. And here I am with other members of CMIC, ending my 20-year hiatus from bicycles to do the London to Brighton bike ride. Most of the fellowship involves software development for the lab or optimizing imaging analysis pipelines. And this is where I first had my interactions with Nick Fox and Joe Barnes from the Dementia Research Center. Derek offered me a role at his startup company, Ixico, and I decided to give industry a try. This turned out to be actually an excellent decision. I saw firsthand the challenges of using MRI in large-scale studies in dementia, and it was a challenge that I really enjoyed. I set up the image acquisition, quality review, and image analysis for studies in Alzheimer's and other diseases, including here at UCL, where I worked on Sarah Tabrizi's landmark TRACK-HD study in Huntington's disease. It was a job that took me all over the world, working on studies from Beijing to Buenos Aires. But after 5 years, I was missing research. The more I collaborated with Nick Fox and Sebastian Orstelan on projects, the more I realized I wanted to come back to academia. But the time in industry was formative for my research. It helped me identify what I really wanted to work on when I came back to UCL. And that research was how we could use imaging to better understand the decades-long process where Alzheimer's disease begins to build up before any symptoms are present. This is what I'm referring to as the long silent prelude in the title of my talk. For those in the audience less familiar with Alzheimer's disease, I'll briefly try to explain what's happening in these early stages. So for individuals and families living with dementia, the first thing they'll notice is that they're not remembering things as well as they used to, or that they notice other problems with their mental abilities. In the clinic and in research, we can measure these changes in memory and other thinking skills using various tests. At what point these changes represent an official onset of symptoms is often uncertain, as these measures have a fair amount of variability, and there are a range of values that could be considered normal, especially when we're thinking about age and IQ and education. But since Alzheimer's disease and other forms of dementia are progressive diseases, the symptoms tend to only get worse with time. But long before symptoms, several disease processes are happening in the background. The earliest sign of AD is the appearance of amyloid beta plaques in the brain. Amyloid beta is a protein that naturally occurs in the brain. However, sometimes this amyloid beta protein is produced with a different three-dimensional structure than usual, which we refer to as being misfolded. And these misfolded versions of the protein are sticky, and they clump together to form these plaques. Using various techniques, we can detect the buildup of amyloid plaques as early as 20 years before when an individual would experience symptoms. But the second major hallmark of Alzheimer's is the development of another misfolded protein called tau. And tau also normally appears in the brain, where it's usually involved in stabilizing parts of the brain cell. However, when misfolded, tau also starts to stick together and form what are called neurofibrillary tangles. And these are toxic to brain cells. In most cases, tau tangles only spread through the brain once amyloid plaques are present. And the presence of both amyloid plaques and tau tangles dramatically increase the risk for developing Alzheimer's-related symptoms, even compared to those individuals who have amyloid plaques only. So determining how long it takes to go from being just amyloid positive to both amyloid and tau positive is of high interest to the field. These different disease processes start to damage brain cells and impair their function. Eventually, brain cells start to die, and the brain shrinks much faster than it does in normal aging. We can detect this increased rate of shrinking of brain atrophy using MRI scans somewhere around 3 to 5 years before symptoms. And it's only after decades of plaques and tangles building up, causing sustained damage and loss of brain cells, that these symptoms finally become apparent. The good news is recently there have been some breakthroughs in treatment where 3 new drugs have been approved in the past few years. These drugs remove most, if not all, of the amyloid plaques from the brain in as little as 6 months. But they only seem to provide a modest benefit in terms of slowing declines in memory and activities of daily living. These drugs have only been tested in individuals who already have symptoms, represented by this red region in the plot. But many of these drugs remove amyloid, which happens at the earliest parts of this process. So perhaps we should be treating individuals earlier in order to provide the most benefit. What if we could use these drugs to remove amyloid at a much earlier stage so that we can preserve memory and other mental activities as much as possible? Trying to treat the disease earlier does have some major challenges. First, individuals at this stage of Alzheimer's disease have no obvious symptoms, and so any changes to their memory or other mental activities will be very subtle and more difficult to detect. However, levels of amyloid plaques and tau tangles are changing substantially during this period. So the key question will be how to measure the benefit for patients of removing amyloid and tau if they have no symptoms. Will we be able to slow or even stop the onset of symptoms down the line? So my work focuses on how to detect early changes in Alzheimer's disease and other dementias using brain scans. We can measure the amount of amyloid plaques and tau tangles in the brain using positron emission tomography, or PET. In PET, a small amount of radioactive dye is injected into the patient, And some of these dyes attach to amyloid plaques, while others are designed to be more specific to tau tangles. Now then, the subsequent damage that amyloid and tau cause to brain cells can be measured with magnetic resonance imaging, or MRI. MRI can capture high-resolution scans of the brain's anatomy so that we can quantify how fast the brain is shrinking. These scans can also capture other damage to the brain, such as small vessel disease, reduced structural integrity, and decreased use of key resources. So with any of these techniques, if we can make the measurement more accurate and more precise, then we should be able to detect smaller and earlier disease-related changes. One key example of how we can improve these measurements was one of my first projects here at the DRC. I worked with Mark Modat and George Cardoso on some new automated measurements of the hippocampus that they had developed. The hippocampus, which are the structures in blue and red that you see here, are brain structures that are affected early in the AD process. So the new approach substantially reduced both the error in the measurements as well as the time needed to review the results. And once it showed promise, we worked with the DRC Imaging Core Lab, Casper Nielsen and Nia Malone, so that we could incorporate these measures in all of our studies. And in the meantime, George and Mark further extended this technique to not only identify just the hippocampus, but hundreds of brain structures. And together, these techniques are now core analysis pipelines at the DRC, which we have used on thousands of scans across our studies. And so that project's a great example of what my role has been here during my career at UCL. I use my engineering background to work with people from various disciplines and bring novel solutions into clinical research. So I'm going to give a whistle-stop tour highlighting through some of the various research projects that I've led or been a key contributor on, where we use imaging to look for pre-symptomatic changes in Alzheimer's and other forms of dementia. So finding individuals without symptoms, but who do have biological evidence of Alzheimer's, is a big challenge. There are a few ways to address this. One approach is to recruit individuals from families that carry very rare mutations known to cause dementia. So it's virtually certain that someone who carries this mutation will go on to develop Alzheimer's. On the other end of the spectrum, we could cast a wider net in the general population of people in their 60s, 70s, and 80s, but the percentage of individuals at the early stages of AD will be much lower. So I'll start by talking about the research in these rare genetic cases first. These rare genetic mutations are called autosomal dominant, which means if a parent has the genetic mutation, then there's a 50% chance they will pass it on to their child. These genetic forms result in people having symptoms much earlier than typical forms of dementia. Instead of people in their 60s, 70s, and 80s, we see people affected in their 30s, 40s, and 50s. And the DRC is a world-leading center in terms of studying these autosomal dominant forms of dementia, tracking numerous families with these mutations for years, collecting data from family members who have not yet developed any symptoms. The genetic form of Alzheimer's is extremely rare, making up less than 1% of all AD cases. And there are primarily 3 genes where these mutations have been found, but the most common is presenilin-1. However, presenilin-1 mutations can present differently, both in terms of symptoms and in these different imaging presentations. While UCL has a large group of individuals from these families actively participating in studies, we would need more if we wanted to reliably detect subtle changes in the earliest phases of AD. We also would need more participants if we wanted to run a clinical trial to test out promising therapies in this group. So the DRC and other world-leading centers form the Dominantly Inherited Alzheimer Network, or DIAN, bringing hundreds of individuals with this autosomal dominant form of AD into one study. At UCL, we both recruit participants in the DIAN study and analyze data from the entire study. And one of the first analyses I did was to identify what parts of the brain were shrinking in this form of Alzheimer's disease. I searched throughout the brain using a technique called voxel-based morphometry. In mutation carriers who already had symptoms, we found strongest evidence of brain atrophy in the precuneus, a region involved in executive functions, such as planning and organizing tasks, and the posterior cingulate, which is involved in many tasks affected by Alzheimer's. However, the signal was much weaker in carriers who had no symptoms, as indicated by these flatter blue lines in the plots. Many of these carriers were quite young, and so likely too early in the disease process to detect any abnormal shrinking of the brain. As follow-up data on these individuals became available, we were able to measure changes over time within each participant and thus get a better estimate as to when these changes were occurring, particularly during the pre-symptomatic phase of the disease. Working with Chris Frost and Tess Poole, our statistical colleagues from London School of Hygiene and Tropical Medicine, we used this data to model when atrophy starts to become abnormal. And our analysis estimates that this occurs about 3 to 7 years before the estimated age of the expected symptom onset, similar to what other groups found. And it suggests that atrophy is a good proximity marker. In other words, it's a measure that reliably occurs before, but close to the onset of symptoms. So we see atrophy around symptom onset. Can we go a little earlier and detect tau tangles in these genetic forms of AD? I worked with Antoinette O'Connor, who's a clinical fellow at the DRC, to investigate this using tau PET. And since tau tangles usually are detected before atrophy is present, we were surprised to find very few pre-symptomatic individuals with clear evidence of tau accumulation. The regions with the largest accumulation were the precuneus and the posterior cingulate, regions that I just showed were also vulnerable to the early atrophy in genetic AD. And this continued theme of precuneus and posterior cingulate being affected earlier is different than in typical cases of AD, where the hippocampus and other temporal lobe structures are more vulnerable. But we may be able to detect damage in other parts of the brain before atrophy. This means looking beyond gray matter, the areas of the brain cells that process information and make decisions. We also need to determine if there are any changes occurring in the white matter, which is responsible for communicating information throughout the brain. And when you look at white matter on your standard MRI, it's rather uniform and nondescript. But we can use another MRI technique called diffusion-weighted imaging to visualize this complex structure. And my first PhD student, Tom Veal, who was funded by Alzheimer's Research UK, focused on superficial white matter, which is an overlooked part of the brain that is responsible for making small connections between nearby structures. Measuring changes in superficial white matter is difficult, so Tom needed to develop new methods to analyze the data, which we worked on with our colleague, Professor Gary Zhang at CMIC. Now, compared to healthy controls, Tom found that patients with early onset forms of AD had decreased density of neurites in the parietal and temporal lobes, two areas where we also often see shrinking in the gray matter. And the neurites are the main part of the brain cell that makes up the white matter. He did some initial analysis, which suggested two potential causes for these decreases. One is increased levels of iron, something we often see in other diseases like Parkinson's. And the other potential cause is the breakdown of myelin, the insulating layer that helps efficiently communicate signals throughout the brain. In many of these experiments, we were looking at just one imaging measure at a time. But we have so much data covering so many different aspects of the disease. Wouldn't it be useful to look at these all at once and see how they relate to each other? And so this is the premise of the event-based model, which was developed by our colleagues in CMIC, Alex Young, Neil Oxtoby, and Danny Alexander. It looks at all of this data together, determining what measure becomes abnormal first in the disease, then second, and so on and so on. And Neil led our first analysis of Diane with the event-based model and found a pattern where amyloid plaques and measures of tau were abnormal first, about 15 years before expected onset, followed by increased atrophy and decreased function and memory. So when we combine all these results from these studies in genetic forms of AD together, we can detect amyloid very early in the brain. The findings are mixed as to when we can detect tau tangles, and we can detect atrophy closer to when carriers will experience symptoms. So another form of dementia that has these genetic mutations is frontotemporal dementia, or FTD. So it's far less common than AD in the general population, but genetic forms represent about a third of all cases. FTD is a very heterogeneous disease. Most individuals have behavioral problems, but other forms of FTD include difficulties with language, balance, and other motor function. And there are three genes where most of the FTD mutations occur: C9orf72, progranulin, and MAPT. So just like with Diane, it was important to establish a collaboration of expert sites around the world to get a larger pool of participants. And Professor John Rohr from our group led the formation of this worldwide study known as the Genetic FTD Initiative, or GENFI. And we launched the study in 2012 with 12 sites from the UK, Canada, Italy, Netherlands, Portugal, and Sweden. After 3 years of showing it was feasible to collect this data from multiple research groups, we began a second phase that expanded to 26 sites and recruiting over 1,000 participants. And more recently, we have launched a third phase where we've included even more sites and countries and continued to collect data for over 1,000 participants. Now, GENFI was our first time being the lead site of a global multi-center study. So I worked with Dave Thomas, who's a fantastic MRI physicist. I believe he's somewhere in the air on his way to Honolulu for a conference right now. And together we set up the MRI imaging protocol for GENFI, ensuring that all the different scanners across the study were producing similar quality data. We also needed a robust platform to receive, store, manage, and share data. And together with Seb and Mark and a fantastic programmer named Miklos Espak, we customized an open source data management platform called XNAT for this purpose. This provided all the sites a secure web portal to upload and access their data. So over the 13 years of GENFI, we have collected nearly 5,000 MRI scans. And we've used XNAT to make public releases of the data for researchers to use in their own work, generating around 90 publications to date. Now with GENFI, we were one of the first groups to use this platform at XNAT, at UCL, and it's now been widely adopted at UCL, and it's supported by the Advanced Research Computing Center, who supports over 80 studies at UCL using XNAT. It's also used by many groups worldwide. So I led the organization of a large in-person workshop on XNAT, which we hosted here in London near St. Paul's. And it brought together 90 researchers from 10 countries, as far away as Australia, to be a part of it. So what have we learned about FTD from the GENFI study? Well, initial analysis of brain volumes in GENFI suggests that areas shrinking most in FTD were the insula, the temporal lobe, and the frontal lobe. And these regions are known to be vulnerable in frontotemporal dementia, as the name would suggest. These results suggest that the earliest changes might be occurring roughly 10 years before patients are expected to experience symptoms. When comparing the different genetic mutations that cause FTD, we found that each mutation has some unique atrophy patterns. However, there was also a large overlapping pattern in this light, which is this light yellow bit, which is across all 3 genetic mutations, suggesting a common signature of FTD. And the brain regions in this common signature are all part of a functional brain network called the salience network. Now, this network is involved in tasks that require attention and self-regulation of behavior. However, as with autosomal dominant forms of Alzheimer's, pre-symptomatic changes were much smaller and more difficult to detect. So as a result, we wanted to better understand what was driving atrophy in these areas. So I worked with my colleague Andre Altman at CMIC, who explored if there were unique genetic patterns in the brain regions that were most affected. And Andre found that there was likely a strong involvement of astrocytes in these regions. And astrocytes are support cells in the brain that when disease is present, they begin to react, and that reaction eventually becomes toxic itself. He also found involvement of genes that would suggest impairment of the blood-brain barrier. And this blood-brain barrier is the protective barrier that keeps unwanted substances out of the brain. And Martina Boqueta, not only an amazing researcher, but also, I might add, all-time best person to share an office with, used GENFI data to explore atrophy patterns further, investigating deeper structures of the brain termed subcortical nuclei. Among other tasks, these nuclei are involved with inhibition, impulse control, reward, and motivation, all elements commonly affected in FTD. She found that while there are some common regions involved across all 3 genes, the earliest pre-symptomatic involvement was primarily in C9orf72 cases. These cases tend to have longer and slower progression. Again, we also collaborated with Alex Young and Neil Oxtoby to use disease progression modeling to examine these atrophy patterns and determine which brain regions start to shrink first in genetic FTD. But since we know that this disease is heterogeneous, Alex and Neil extended their approach to allow for different subtypes within the disease, each with their own distinct ordering. We found 4 subtypes, one predominantly of progranulin carriers, one of MAPT carriers, and 2 different subtypes of C9orf72. And these group of C9orf72 carriers have been known to present with different symptoms and slightly different disease processes. So overall, genetic FTD is rather a heterogeneous disease with different atrophy patterns going on, but there are some commonalities which might help when we start to begin treatments in these diseases. So I've highlighted the work I've been involved with in presymptomatic AD in these unique genetic cases. So now I'm going to switch gears a little and talk about what we can learn about the early stages of the disease from the population studies in older individuals. And this work focuses on the amazing Insight 46 study led by Professor John Schott, a study that I've had the fortune to be involved with since it started. So no matter what my kids say, I was not around at the time to remember what Britain was like in 1946, but it was obviously a very different place to what we experience now. My children might be working instead of attending school, and things we now consider essential were not common back then, and in some cases they were even a luxury. It was also a time when the UK was experiencing a worrying decline in birth rates, as well as high rates of infant death and poor health in children. Scientists and doctors were extremely concerned about this and wanted to investigate the underlying causes. As a result, they created the National Survey of Health and Development and sent it to all parents who had a single baby born during 1 week of March 1946. And from this initial survey, there were some clear messages in terms of the high cost of childbirth and the provision of pain medication to mothers during labor, which led to substantial policy changes. Now, to also illustrate what a different time this was, John found this astonishing question in the original survey, asking the mothers who looked after their husbands while they were in bed with the baby. Now, I personally dread to think what would happen to anyone who asked this question to any of the women in my family. But this study did not stop with that single survey. Many of these children were followed for their entire lives with over 25 waves of data collection, from childhood IQ tests to questions about shift working to their physical activity and blood pressure measurements taken during midlife. And as they were approaching their 70th birthday, it seemed like an ideal time to start to look for early signs for Alzheimer's disease. The overall prevalence of AD at 70 is quite low, but the number of individuals with amyloid plaques in their brain is much, much higher, around 20%. This means that there should be several participants in the study who are in the earliest stages of AD. So we recruited a subset of these participants into a study called Insight 46, and we acquired PET and MRI brain scans designed to spot changes in brain health as well as signs of presymptomatic AD. When we first started preparing for the Insight 46 study in 2015, I started to map out the data flow and how everything would fit together, and it resulted in this insanely complicated diagram, which highlighted some of the challenges that were involved in the study. But thankfully, from GenFeed, we already had a great team in place. So Dave Thomas and I worked on the imaging protocol. We worked with Dan Beasley, who focused on data management and transfer from the hospital. And Mark Modat led the design of the imaging analysis pipelines. And then I, along with colleagues from the DRC, processed all this imaging data. And the results that we have generated from this data have played a key role in over 25 publications on Insight 46 data. For Insight 46, we recruited 502 participants for the main study. And since all the participants were born within 1 week, our age range was extremely small. And the left-hand map shows where these participants lived, which is reflective of the overall UK population, and you see on the right-hand side. So we would expect to find some people with evidence of amyloid plaques, but how many? And this was work I did with Will Koth, who just finished his PhD and is now in Madison, Wisconsin, where we used a technique called Gaussian mixture modeling. And that identifies 2 different groups from this data. The first group are individuals in this large curve on the left who are likely amyloid negative. The second group, represented by the flatter curve on the right, are the ones who are more likely to be amyloid positive. And from this model, we estimated approximately 15 to 25% of these unimpaired participants who had amyloid at age 70. Much of this variability arose from how the data was processed, highlighting that there is some uncertainty when using a single value to determine whether someone has a negative or a positive scan, especially in the earliest stages. However, it's difficult to compare different flavors of processing amyloid PET data, as they have different ranges and different levels of error. As part of his thesis, Will successfully calibrated all of our amyloid PET data to a universal scale known as centiloids. And centiloids are a Celsius-type scale where 0 essentially means no amyloid, and 100 represents the average level of amyloid that an individual with AD would have. The consensus across many studies is that the range of 10 to 30 centiloids represents the initial signs of a positive amyloid scan. And for the Insight 46 study, we found cut points slightly on the earlier side of this at-risk range, from 12 to 19 centiloids. Once Will and I had generated this data, researchers could use it with all of the data collected throughout the life course of these participants. So we could essentially look back in time and see what factors from childhood or midlife may be related to amyloid plaques or smaller brain volumes of age 70. And an excellent example of how this works in practice was Chris Lane's work investigating potential links between heart disease and dementia. During his PhD, Chris looked at a number of measures during his midlife that were related to heart disease. Even at age 36, he found associations between increased risk of heart disease based on a measurement called the Framingham Risk Score and lower brain volumes 3 decades later. He also found that increased risk for heart disease was associated with increased white matter hyperintensities, a sign of vascular injury in the brain. However, there was no evidence of a link with measures that were more specific to Alzheimer's disease, such as amyloid PET or hippocampal volume. Chris also performed a similar study using blood pressure measurements from midlife. Again, a similar pattern emerged here. Increases in blood pressure were associated, and changes in blood pressure over a decade were associated with lower brain volume, lower hippocampal volume, and higher white matter hyperintensities. But again, no relationship with amyloid. Together, these findings suggest that long-term risk for heart disease can affect brain health decades later, but it does not result in a clear evidence of increased risk for AD. But what it could do, this damage to small blood vessels may combine with amyloid to increase how quickly the brain shrinks during the pre-symptomatic stage of AD. So Sarah Coyce, a PhD student who I worked with on Insight 46 data, found that white matter hyperintensities and amyloid plaques provided independent and relatively equal contributions to brain atrophy. And this finding has been seen in other studies from Harvard and also a large multi-center study in work done by my colleague, Joe Barnes. And one of the most recent and exciting breakthroughs in AD research is the development of simple, accurate blood tests to detect plaques and tangles in the brain. The blood tests could potentially revolutionize early diagnosis, as it's far cheaper than a PET scan, and it does not involve injecting any radioactivity into the patients. But these tests also need validation. So we provided the amyloid PET data to Ashwini Keshavan, a clinical research fellow working on these blood tests in Insight 46. And she determined that these blood tests were picking up the same individuals who were amyloid positive as the PET scans. After the first wave of imaging in Insight 46, we invited the participants for a second follow-up scan, roughly 2.5 years later. Now, in general, the level of amyloid either stayed the same or increased over time. There were no individuals who went from amyloid positive at their first scan and returned to normal in their second scan, represented by this empty blue region in the bottom right. We did, however, find roughly 8% of individuals who were amyloid negative on their first scan and then developed amyloid and became amyloid positive 2.5 years later, this upper left region in the green. And this is related work from Ariane Balak, who did her PhD with myself and Frederick Barkhoff, and she investigated what level of amyloid increase over time would be considered outside the margin of error, and thus a reliable signal of the disease. And she found that these reliable rates were in the range of 2 to 4 centiloids per year, based on results from both the Insight 46 data and another large European cohort. We can then take these two measurements, the initial level of amyloid and the speed it is accumulating, to estimate the age when an individual first becomes amyloid positive. To kind of illustrate this, when we plot how much amyloid somebody has in their brain with respect to their actual age, it's a mess. We don't see any clear signal here. But by using a technique called SILA, which was developed by a good friend of mine, Toby Bethauser at the University of Wisconsin, we can align all the participants on a single amyloid timeline, and the progression becomes much clearer. Positive values of amyloid duration represent how long an individual has likely had amyloid in their brain. And negative durations are our best guess as to when an amyloid negative individual will become positive. So in Insight 46, despite having a very small range of ages in the participants, the range of where these participants sit on the amyloid timeline is almost 4 times larger. And so to illustrate this more, this movie shows a range of Insight 46 participants moving along this amyloid timeline. The cool blues and purples represent very little amyloid in the brain. And we start with participants with no amyloid present and are likely decades away from having signs of plaque. But as we get closer and past onset, you begin to see more warmer colors, greener and hotter yellows and reds, indicating the buildup of amyloid. And once we are past onset, you can see dramatic progression as more and more amyloid builds up in the brain, until we get to some individuals who have 100 centiloids, which is the same amount of amyloid plaques that you'd see in patients with Alzheimer's. So Will and I investigated what factors might alter this timeline. He found that the most common risk gene for ADE, called ApoE, shifted this amyloid timeline about a decade earlier, which is likely why people who carry this gene tend to have symptoms at a much earlier age. And there's also suggestion that female carriers of this gene might have a slightly earlier amyloid onset than men. After scanning Insight 46 participants twice with this amyloid PET, we then invited some of them back again for a third scan when participants were aged 75 to 78. But this time, we're measuring the levels of tau tangles in the brain using PET so that we could get an estimate of the timing between amyloid and tau. So we found a handful of individuals, definitely less than 10%, who had tau and tangles present, but no evidence of amyloid plaques from their earlier scans. And as they started to show evidence of amyloid and started to build amyloid up, we saw that the percentage of tau-positive scans roughly doubled in those individuals. And this increased even further for those who had the greatest levels of amyloid burden. So these results suggest that tau tangles are not usually present until there is first a sufficient buildup of amyloid plaques in the brain. So if tau is not present until after amyloid, can we estimate the delay between amyloid onset and tau onset? So just like with amyloid, when we plot tau with respect to the participant's actual age, again, we don't see a particularly clear relationship. However, when we plot against this amyloid duration, we get a much clearer picture where all the tau-positive scans come after the onset of amyloid. So we can then use some statistical models to estimate how long after amyloid onset the tau starts to accumulate. In regions where tau is known to accumulate first, the gap between amyloid and tau appears to be about 2 to 3 years. In regions that tend to accumulate later, when the patient is closer to experience symptoms, that difference is about 10 to 12 years. So this work from Insight 46 has really given us a better understanding as to the timing of different disease processes during this long silent prelude of AD. These imaging measures are well established and widely used in clinical trials as an outcome measure to determine whether a drug is effective or not at slowing down Alzheimer's disease. So I've done a number of projects looking at how we can use imaging in clinical trials, many of them with Professor Chris Frost from the London School of Hygiene and Tropical Medicine. Chris is a brilliant and very patient biostatistician who has taught me so much during my time at the DRC, and he's helped me develop a deep appreciation of statistics that I certainly did not have from my introductory undergrad courses. When designing any clinical trial, we need to know how many participants are required. If too many participants are enrolled, the trial will likely take longer and cost more. And it's also wasteful and potentially unethical to recruit far more individuals than needed. Therefore, we want to keep these sample sizes in these trials low, but not too low, because if we include too few participants, that would mean that there may not be enough sufficient statistical power to detect that a drug's actually working. It may also mean that results will not hold when applying them to the broader context of people with this disease. So for any proposed trial design, we can estimate the appropriate sample size required to detect the desired treatment effect that we think would provide sufficient benefit to individuals with this disease. And together with Chris and his team, we had estimated sample sizes for imaging measures over a number of different trial scenarios. And my first sample size analysis with Chris was also with Seb Orsolan, Ian Malone, and John Schott, where we hosted an open science challenge. Now, these challenges are essentially competitions between researchers. And the goal of this challenge was to produce the best measure of brain atrophy for a clinical trial in AD. We provided all the teams with exactly the same imaging data, and then we analyzed their results centrally. We found the estimated sample sizes from each submission. Many methods produce comparable results, with the best performing methods providing sample sizes on the order of 120 to 190 participants to detect a slowing of brain atrophy by 25%. With the ventricles producing the most common sample sizes, as indicated by the relatively tight confidence interval lines you can see on the plots, with the most uncertainty arising in the hippocampal measures. We also saw similar estimates in Elizabeth Gordon's work estimating sample sizes for trials in frontotemporal dementia. One of the key findings from her work was that direct measures, where we took a look at both scans together and look at the change between two MRI scans, perform better than if we did a measure in one image scan and then we did a measure in another image scan and we subtracted the difference. But we've learned a lot over the years about what's actually needed to produce a benefit meaningful to the patients. And the results from recently approved drugs suggest that reducing the speed that the disease is progressing may not be good enough to provide meaningful benefit to the patients. And this is especially true when we are talking about removing amyloid plaques, where the key to producing any sort of meaningful benefit to the patient is to remove a substantial amount of amyloid over a very short time frame. So for measures like brain atrophy or memory, measures which reflect damage that's likely irreversible, we still should define effectiveness in terms of how much we can slow the disease down over time. But for measures of plaques and tangles, which these drugs are designed to try to remove, we need not just to reduce the speed, but we need to reverse the direction entirely and to find a successful treatment in terms of how close we can get these measures back down to a normal range by the end of the study. So we applied these proposed treatment effects to look at presymptomatic trials in autosomal dominant forms of AD. So how many presymptomatic carriers will we need for a new trial? And this is particularly important in this form of AD, since these cases are so rare. For outcomes involving memory of atrophy, we would need at least 80 to 200 participants per arm. But notice the width of these various lines. They represent the range from the best case scenario to the worst case scenarios for each outcome. So for some measures, the uncertainty was very high. So while the best case scenarios looked appealing, the worst case scenarios could require thousands of participants. For rapidly removing large amounts of amyloid or tau, which is a pretty large effect admittedly, we see that the sample sizes are much lower. With roughly 50 participants per arm, with the worst cases reaching up to 150, we should be able to detect the minimum level of amyloid removal that we could think could provide some benefit to the patients. So that covers most of my research, but outside of research, I've been involved in a number of educational activities. So working with a number of groups at UCL, we created an initiative called Health and Bioscience Ideas. And this program developed various hands-on interactive training activities for researchers who wanted to use data science and image analysis techniques in their work. I've also been heavily involved in education through the Alzheimer's Association's Neuroimaging Professional Interest Area, a professional organization of over 1,700 researchers specializing in neuroimaging and various forms of dementia. We've worked hard on our educational outreach to our members, including educational webinars, some in different languages for making it more accessible to researchers around the world. We've developed hands-on neuroimaging workshops that anybody can do as long as they have a laptop and a web browser. And the best part of it all is that along the way, I've made some lifelong friends. We've enjoyed working together on all these projects. So I thought I would close by taking a few minutes to describe my future research plans. My first goal is to continue research on how imaging can be used in clinical trials of pre-symptomatic AD. As I discussed earlier, detecting small changes in memory scores during the stage will be difficult. It will require thousands of participants and following them for over many years. From our sample size estimates, we know that it would take far fewer participants to detect substantial reductions in amyloid or tau in a clinical trial of pre-symptomatic AD. However, we need further evidence that these reductions in amyloid or tau would delay the onset of symptoms. So the Alzheimer's Society has generously supported me with the Dementia Research Leaders Fellowship to continue this work in a project called NOPAD, which stands for Novel Trial Endpoints in Alzheimer's Disease. The first step is a massive data initiative to collect imaging and other biomarkers on unimpaired individuals across a number of global studies. I'll then analyze the data to estimate rates of change and long-term risk of developing symptoms from measures like amyloid and tau. And finally, I'll work with my colleague Katie Morgan from the London School of Hygiene and Tropical Medicine to use these results to explore new clinical trial designs involving pre-symptomatic individuals so that we can find better ways to run these trials during this pre-symptomatic phase. The other area I want to focus on is how to make these neuroimaging techniques more available and accessible. Alzheimer's disease affects individuals from all ethnicities and backgrounds. But often our studies do not look like the general population in the US or the UK or in other countries. And this will have a knock-on effect when it comes to access to treatments. So while I was beginning to think more about this problem, I was invited by Adam Smith to be on the Dementia Research podcast, where I had a fantastic conversation with Shana Stites from the University of Pennsylvania. And Shane is an Alzheimer's researcher who focuses on social determinants of health. So after the podcast, we continued this discussion by organizing a workshop where we gathered many leading experts in both neuroimaging and diversity and health disparities, and we drew up a number of solutions for how we could make neuroimaging more accessible to more potential participants in clinical studies and clinical trials. And we're now currently drafting the results from these discussions that we're going to provide in a paper. One approach that may help is the use of ultra-low field MRI scanners. These scanners are more portable, use less power, require less technical support, and are less claustrophobic to the patient. So thanks in large part to James Cole, we now have one of these scanners at UCL. And my PhD student, Aaron Jozic, will soon start scanning FTD patients using the scanner. We're also hoping to use ultra-low field MRI to study children and elderly individuals in Guayaquil, Ecuador, a region that's been severely affected by both extreme heat from climate change as well as exposure to pesticides from nearby banana plantations. So this would be looking at a birth cohort of individuals that were originally followed 8 years ago, and since most of these households are multi-generational, they have a lot of grandparents as well. And so the idea would be this would be one of the first studies to look at what effects climate change have on brain health, looking over a multi-generational perspective. Now, while these images are nowhere near the quality of a conventional MRI, they still provide useful information and can help us address new research questions. So, it's been an honor and a privilege working with such talented individuals throughout my career. So I just wanted to thank a few more people who have provided me support over the years. I want to thank my mentor at UCL, Professor Nick Fox, for always being so supportive of my research and for championing my work. Your input has always made my work better, and to have an inspiring, incredible researcher believe in me has meant a lot. I'd also like to thank my PhD supervisor, Bob Galloway, whose door was literally always open to talk, whether it was about image processing or basketball, to Seb Orsalan for his help championing new biomarker discoveries, our work with XNAT, and all your efforts to help us translate these new developments into the clinic and research. And to John Schott and to John Rohr, thank you for all the support over the years. It's been incredible being on the ground floor of studies like GenFi and Insight 46 and watching them see how they develop. To the brilliant PhD students I've been fortunate enough to work with on their research. One of my most favorite parts of the job is talking with you guys every week about science, your projects, and everything else. Thank you to all the neuroimaging colleagues that I've enjoyed working with on these projects day in and day out. And finally, to my brilliant family. To my parents and grandparents, who provided me a loving, stable household and unending amounts of support, belief, and cheering on. To my in-laws, Peter and Sydney Charles, who have provided so much support ever since I moved to the UK. To my amazing wife, Helen. You know, I work in an institution that has produced several Nobel laureates, but I really don't know anyone who's more brilliant than you. Throughout the years, you provided me support on my down days, laughter, fun, and adventures, and you have always motivated me to continue to improve. I love you very much, and you will always, and have and always will be, the north on my compass. And finally, thanks so much to my fantastic kids, Claudia and Charlie. I love you guys both so much. Very little else matters when you guys are around. I have so much fun with you, and I enjoy and cherish every single second I get to spend with you. I love my research in brains and Alzheimer's disease, but what I find far more fascinating are the journeys that you'll go on in your life, and I can't wait to see what they are. So I'd like to acknowledge my funders for their generous support, particularly to the Alzheimer's Society for supporting many of my research projects, and to everyone else I have had the fortune to work with over the years. Apologies that I can't single each of you out during this talk, but I'm looking forward to celebrating with you at the drinks reception after. Thanks very much.
Well, thank you, Dave, for a wonderful lecture. Not only were the science and the research contributions clearly excellent, but it beautifully presented, and I wonder how much the time as a radio DJ had influenced your ability to present. I mean, those who regularly tune into 91 Rock may well have picked up some of the features there. But I am still intrigued, Dave, about what the soundtrack to your lecture would be. Not after the silent prelude, what comes next? But maybe that's for another evening. But on a more serious note, I want to acknowledge the huge amount of hard work and dedication that goes into this journey. It doesn't get achieved lightly, not just putting the slides together, but to be awarded tenure and the title of professor. And I think you showed us all just how much you've brought to the field as an engineer, as a biomedical engineer. And in preparing for this vote of thanks, I looked up the definition of engineer, and there are many. One related to using scientific knowledge to design, build, and maintain engines, machines, or structures. Another definition, to skillfully arrange for something to occur. And I think Dave's work and his lecture demonstrate just how effectively he has done that. He has designed, he has built, and he has maintained, and very skillfully arranged for critically important studies to occur and to deliver effectively. But I think these contributions are not sterile, arcane, not just about papers and contributions to science. Dave's contributions are fundamentally translational. They're about making a difference to patients. And it's important not to lose sight of that when we list the hundreds of papers, the millions of grant income. But Dave's contributions are, yes, they are about methods. And yes, they are about improving our understanding of science and of these diseases. But they are very much about making a difference, about improving diagnosis, making clinical trials that are so important, just that little bit more effective, run better, so we can find the desperately needed treatments for these devastating diseases. And Dave, while you are clearly very good at putting things together, as a good biomedical engineer should, you're also very good at putting or pulling people together. You are hugely liked, and you are hugely respected by colleagues around the world, and nowhere more so than here at the DRC. It's actually quite difficult to find anybody to have a bad word to say about Dave. I've really tried. I've looked around. I've the quotes. And I think, without being too philosophical, just as I'm the few minutes between you and the drinks, it's actually something, that ability that you can achieve collaboratively, that it's not just a zero-sum game, it's not just about getting the best part of a deal, is very important in this very turbulent world we have at the moment, and where conflictual contracts are increasingly the norm, rather than, as you demonstrate, the fantastic teamwork that makes a difference, and how you demonstrate not just your clear affection for those who are closest to you, but your kindness, your generosity to students, your work as a graduate tutor, but just as a great colleague. And so I think I speak for many, but definitely personally, not just for how important you are to the success of the Dementia Research Center, for the imaging pipelines, for these huge studies, but your contributions to the DRC, your kindness, your generosity, your good nature, and your friendship. Please join me in thanking Dave for a lovely, wonderful lecture without a soundtrack.